So our second speaker is Dr. Maria Asplund from Freiburg University in Luleå in Sweden. So we're very happy that you could join us today. So please.
Thank you very much for this invitation and for the opportunity to come and meet all these nice people, hear all the talks, and also to tell you a little bit about my research that I do at the University of Freiburg in southern Germany. So my name is Maria Asplund. I've been working in Freiburg for the past 11 years. And I work exactly on these questions, now how to stimulate with greater precision. So today I decide to tell this story not focusing so much on the peripheral nervous system, but actually the technologies, they are directly applicable to those kind of questions as well. So I would like to talk most of the time about how to restore vision with visual cortex implants. And then if time allows, I would also like to say a few words about regenerative stimulation and how we can use stimulation, hopefully, to improve spinal cord regeneration or even skin regeneration in the future. So, bioelectronic vision, that is a concept that's it's a really old idea. So the idea is that if you have someone that is blind, you can use stimulation, a pattern of stimulation at various levels in the visual system to induce artificial vision. So this could be a picture from the camera translated to a pixelized stimulation pattern that could be injected either at the retina, at the optic nerve, in the lateral geniculate nucleus, or directly into the visual cortex. So all of this is possible, and experimental work is ongoing on all these levels. So I would like to focus today in particular on the visual cortex, because that is the technology that has the potential to address the largest population of blind individuals. Of course, the implants are going to look quite different. If you address the retina, we need some kind of matrix for stimulation, but in the visual cortex, we need to go into the actual brain. So we need penetrating probes for these kind of tasks. So, bioelectronic vision, I told you it's an old concept. I wonder how many of you know that the first reports of these electrically triggered phosphine, it dates back to 1755. So it's even before Galvani phrased the concept of bioelectricity. It's older than that. So the main question one should ask is, after more than 250 years, why am I optimistic that somehow my research team and my collaboration partners will be able to finally solve this challenge? So hopefully I will be able to tell you a little bit about why I'm optimistic. So if you take a look at bioelectronic vision today, so what can already be done today? So most of the work on visual prosthetic systems, they are performed with exactly these kind of arrays that we saw before, so the Utah electrode array. So you push these electrode pins into the visual cortex. And most likely you need many of these devices to cover a larger area of the visual field. And then you stimulate in different patterns. So I have here a small movie from our collaboration partners in the Netherlands. They do this with the Utah array, and they have here a simple experiment. And we see here the monkey he receives a stimulation pattern, so that's represented on the screen here as the white spots, but he actually doesn't see that. He only gets this as electrical stimulation in the visual cortex, and he's supposed then to report on if it was an A shape or an L shape. And if we look at this for some time, we can see that most of the time he actually gets this right. So these kind of experiments are able to tell us something about how well this concept works. So pattern stimulation in the visual cortex, can we build complex shapes with that? However, there are many problems, but the main two is the first one is the limited resolution. So it's very difficult to make these devices smaller so that we can stimulate only few neurons and get much larger, be able to build much more complex pictures. And the second one, and that has really been what's keeping us from doing this at larger scale, is the lack of stability over time. So not only are these silicon-based devices rather unstable in themselves, so the electrodes corrode over time, but also the scarring in the brain prevents close contact between device and nerve tissue. So after, let's say, 1 or 2 years, there will probably be no residual function of such a cortical interface. So, why is this? Well, ideally we would like to make extremely small electrodes, so much smaller than the Utah electrode array. And we would like to have these electrodes in intimate contact with neurons, so we can stimulate only few neurons at a time, which means that the threshold current we need to inject to trigger a phosphine remains low. In reality, scarring comes in, meaning we will have inflammation, we will have a glial scar forming, which is an insulating capsule. Some neurons close to the device will die because of the neurotoxic environment. We will see phosphines going up. The threshold to inject phosphine will go up, so we will need to use more and more current, which puts our small electrodes at risk of corrosion. So most likely we will not only damage the device, but also generate electroactive species like reactive oxygen species, or change the pH. So we will have even more inflammation and even higher thresholds. So this is usually what happens. Fortunately, we have learned really a lot over the last 10 years. There's been so much progress in trying to understand what we need to do to address this problem. We have learned how to make much more stable probes. We have learned how to make more efficient electrodes so we can push more current out before we reach the limit for corrosion. And we have started to understand what we need to do to get this tissue interface stability. And first we were very much focusing on the cell surface interaction, so thinking we should make softer surfaces or have some kind of drug releasing interface that would treat inflammation. But what we have come to understand is that the key is really structural biocompatibility. So structural biocompatibility means the biomechanics, not just of the material, but of the complete device. So I'll explain that with some example data from epicortical grids that we made. So these epicortical grids, it's for another purpose, but it's a soft, flexible grid that is placed on the surface of the cortex to record superficial neurons. This is made in polyamide with platinum metallizations. And it's supposed to be able to record over many months and induce little scarring. That's the whole idea. And simply by changing the thickness of the polyamide, of the polymer construction material, from 12 micron to 8 micron, we can see, I don't know if you can see it here, I'll try to point it out. Here we see there is cortex depression with the thicker devices, but when we start to thin the devices down, we can see that because the device conforms to the brain curvature, there is actually very little scarring. So picking just a flexible material is not the full solution to the problem. We also need to consider the geometry of the device, and of course also the interconnection, so tethering forces also play into this. So using the same materials, the same microfabrication technologies, we have made a large number of different devices. So this is currently what we have, our state-of-the-art device. So this is a multi-layer assembly. Each yellow film here is polyamide, and in between there's platinum, and then there's conducting polymer electrode materials here to boost the performance. The devices are extremely flexible, as we can see here, so you can really tie a knot with the device without damaging it. And they are also extremely small, so it's smaller than a human hair and still can have just one single shank, 30 electrodes and more on that shank. We have tested these devices for recordings first of all, so placed them in the cortex of mice and recorded neural signals. So for this group in New York, what they wanted was a probe where they can follow signals from single neurons over time. So they want to see the same signature of the neuron, and that shouldn't change a lot over several weeks, so that they can do studies on learning and memory, and that they can really study the neural circuit in detail. So this is what we see here is the same neuron that is recorded at each of these electrode sites. And it's also over a period from, I think it's day 41 to day 63, so we can see that the signal is also fairly stable. So we have up to 6 months of recordings with these devices. We know they survive. So now the question is, of course, can we also use them to solve the tissue integration problems with a visual prosthetic system? So we have, first of all, we have of course, tested these devices a lot in simple saline, pushed a lot of pulses out of the devices to make sure that we understand at which current level they would corrode. So we are now at 10 billion pulses, and we still haven't corroded the electrodes. We know they can survive for very long usage times. And we have also implanted these electrodes. So together with colleagues in Amsterdam and the Peter Rolfsema group that works a lot with visual prosthetics, they have implanted these strands of electrodes into the mouse visual cortex. And this mouse model, they train the mice so that they can report if they see a spot of light. And then they remove after some time the real optical trigger, and they have only electrical trigger. And this way you can investigate which current threshold you need to reach to generate a percept that the mouse can actually then see with this artificial interface. And where we're at right now is that we can see that around 5 microamps is what we need to push out of an electrode to generate one spot of light. And this is well within what these small electrodes can do, which means we have every reason actually to be optimistic that we have solved some of these fundamental problems with long-term stability of these kind of interfaces. So, of course, probes is just one of the items you need to make a visual prosthetic system. I'm now involved in a European project, so I make the probes and electrode materials, and on top of this, there are people, of course, working on smart interconnects. So we aim to have 4,000 single electrodes, which means we will need multiplexing and a smart interface to make this really work. We have colleagues that work, of course, on the visual neuroscience, so understanding how should one stimulate to generate certain shapes. Test models is a big part of this. So historically, most of the visual prosthetic work has been done in non-human primates because you need quite complex models to be able to ask such questions as, do you see an L or an A? But what we try to do is to use more small animal models, which takes actually a lot of work to establish how to do these visual behavioral tasks also in mice. Tracking of eyes and head movement will likely be an important component as well to make this artificial vision in the future function as similar as possible to the real vision. So when we use our eyes, we also integrate information on head position and eye movement and a lot of other things in generating this idea of vision. So the cortex is not just a pattern of stimulation, it actually integrates also a lot of other information to generate the impression that we see something. And last but not least, there's also a team involved that works on how to distribute. If you have 1,000 phosphines to play with, how should one use them to give the most important visual cues to potential users? So possibly they don't want a black and white pixelized vision, but they rather probably want to know things like where the street is, or if there's a tram coming towards you, or where the pavement ends. These are the kind of cues that you really want, and we need a smart use of those phosphines to make a system that is truly useful. So if time allows, I would now like to say a few words as well about regenerative stimulation. So this is basically a similar kind of microtechnology. We can of course make implants of other shapes and for other uses. And one such use, which I think is rather exciting, is to place these implants onto injured spinal cord and to use them both to record from the regenerating tissue, but also to stimulate, to try to use the electrotropic ability of nerve fibers to align when they regenerate in direction with the electrical field. So this is some preliminary data we see here on the slide. This is from our collaboration partners at the University of Auckland. They work with a rat model for spinal cord injury. And we have designed this device that is placed on top of the spinal cord, and we have just then received the first recordings after 3 months. So we can see we can record spikes from the spinal cord even after 3 months. So there's very little scarring. And as next step, we will now start to use these implants also for stimulation, so that we can then see if we can have a beneficial effect on stimulation and which kind of stimulation parameters we should use to get the most effect out. So this project actually is also a little bit a spin-off from what I do in my ERC project. So there I work with electrical stimulation to promote healing of skin. So the idea here is to exploit the natural electrotactic ability of skin cells. So electrotaxis means that cells align their migration with an electrical field, and skin cells, we know from cultures that they tend to do this. And what we want to find out is if it is possible to use this effect to promote faster healing of large or complicated skin wounds. So we have here some cell culture data. This is from single cells. So we can really see here when we increase the field strength. So here we have a 0 field, then we go to 50 millivolt per millimeter, and then up to 200 millivolt per millimeter. And this is a statistical graph showing the direction in which the cells move. And we can see at the higher field strength, more or less all cells move in the same direction. So this is a very well documented effect. And now we are studying if this also holds true once the single cells form epithelium, and once we use a scratching method to make a wound in this epithelium to see if this also makes these artificial wounds heal faster. So I will show you this movie. It's not entirely easy to see actually, but this is the control side. This is the stimulated side, and you see here are the cells, here is the wound in the middle. So here there is no stimulation, here there is stimulation, and if I start this movie, you will see that on the stimulated side, there is a tendency of the cells to faster invade this region in the center. So they really do close the wound faster. And the reason why they come out looking fairly similar is because the field here is not fully directed towards the wound center, but actually it's a channel system where the field direction will be a little bit in the center and then up in this narrow channel here. So this is the reason why we don't see more of an effect, but it's really clear that the cells behave this way. Of course, once we start to play with applications for skin, this means we will have to abandon some of the technologies that we are used to. I mean, it's very comfortable. We work in the clean room. We use gold and platinum and iridium for our devices, which means every device is super expensive. So you can make micro devices very easily, but if you want to make centimeter-sized devices, you need to do something that is cheap and that can be disposable. So to make this possible, we connected to colleagues in Karlsruhe at the Karlsruhe Institute of Technology, and they taught us this cool process. So we use actually the same kind of polymer material, polyamide, and then we take a laser and pyrolyze part of this polyamide material into conducting carbon. So it's going to be a graphene-like material, and on this graphene-like material we then print a conducting polymer hydrogel electrode, which means we can make a complete electrode just made of polymeric materials that can actually be made super cheap. So we have tested these devices a lot for direct current stimulation, which we want to use them for the wound healing assays. But we have also been fortunate enough to connect with Max, and also just as preliminary tests, see how well these electrodes compare to the industry standard of silver chloride for skin electrodes. And I don't know if Max agrees, but I think it's an extremely positive result that even with the first attempt, where we didn't optimize the device actually at all, so we just made a few test samples. The device is still rather thick, it's not really conforming to the skin, but it was still performed almost comparable to silver chloride, both in the bioelectronic pattern recognition and the sensory feedback stimulation trial. So we were quite optimistic that we really could push this further by optimizing simply these materials for these applications. So that's about what I wanted to tell you. I would like to end with really thanking in particular my research group in Freiburg, so all very talented and fantastic people who pitched in on all the practical work. Also, in particular, I would like to name Thomas Stieglitz, my mentor in Freiburg. He is, at least in Freiburg, a little bit the godfather for this polyamide flexible microtechnology. and also Professor Korvink and Professor Busaki, who both pitched in. So Professor Busaki to the recording data that I showed you, and Professor Korvink to this pyrolysis process.
All right, so we have one question from the audience. Do you think cortical interfaces will be able to reliably restore vision beyond navigation?
That's a really excellent question. I think it's all a matter of time. So I think now we know what to do. We have understood where this glial scarring comes from, but I still think there is some technology missing to really resolve it for devices where we need 4,000 electrodes. So it's one thing to make one string of electrodes, it's another thing to implant 100 or 1,000 of those into the visual cortex. I think there will be more scarring because of that. So I think it's theoretically possible, but we will need even better, even smaller probes for that. I think in the next 10 years, I will be super happy if we are able to have some kind of reliable navigation visual system. I think this is ambitious, but to have vision the way we normally experience it, I think this would require 50 years. Maybe I'm too pessimistic there. But one shouldn't underestimate the value actually of having navigational support.
Great, thank you. So I think we come back to the issue of the resolution of the neural interface. Well, thank you a lot, Dr. Asplund.